Okay, so we'll move on to part 3. We're going to talk about our breeding program. So start with some hybridization first. Our WSU quinoa breeding program started in 2010. And when we started, it was with a grant from the Organic Farming Research Foundation for about $15,000. That really helped launch quinoa research at WSU. Back then there was just one person working on quinoa. Now we've got 10 different faculty and 10 grad students all working on different aspects of quinoa, not just breeding, but a lot of agronomy, entomology, food science as well. So it's really exciting. We're a nice tight-knit group. We work well together. We also work with Oregon State researchers there and Utah State University quite closely, in addition to Rick and Jeff from BYU. So we had to start somewhere. I'll talk about some populations we got from Rick and Jeff at BYU, but first we wanted to select our own, populations, really our own progeny, our parents. Which one we did several years of trials, seeing which varieties did the best here, and then throughout we've been making crosses with them. And so compared to wheat, barley, several other crops, quinoa is extremely hard to hybridize. I won't go into too much. There is some tricks to doing that. We're in the process of putting out an extension bulletin on quinoa hybridization. But so here you can see just the initial steps from far left are taking off the leaves one by one, and then just isolating a few of the flower clusters here, and then finally we put it in this tubing, dialysis tubing, for isolation and wait for it to flower. Just as it's starting to flower here, you can see the clusters. They're about ready for emasculation right now. These white lines here show about where we cut each flower cluster, and what that does is allow us to emasculate. So it exposes the anthers and allows us to remove them. As I mentioned before, some of the flowers are female, so if we do see a female flower and don't have to emasculate, we'll automatically go for that. It's much easier. However, that really depends on variety, depends on the environment. There's a lot of things that trigger the formation of females versus perfect flowers, and so this is often what we have to do, is emasculate. And then after emasculate, we'll just, again, in the dialysis tubing, put the female parent and the male parent together and just tap it for a few days, and we get pretty good success from this hybridization here. We do this all through a microscope. So again, wheat and barley we just do with our naked eye out in the field or in the greenhouse. This we have to do entirely under a microscope so we can see what we're doing. So, and then we have to verify to make sure the cross actually happened and there wasn't any selfing. So what we often do is just look for dominant morphological markers, and so what we've done entirely so far is just do crosses with seeds or seed coats with different colors, or leaves with different colors, or big differences in plant height or leaf shapes, anything like that, and that we know the genetic background for, and then we can make sure that there has been a cross. So this picture shows Temuco here, this yellow flowering variety on the left, and Bio Bio, this red flowering variety in the middle here. And then these are two F1 progeny that show, it's a little hard to see in this picture, but they're more pink than they are red, so it's more of an intermediate type. But because the F1 is large, the red seed coat color is largely dominant, we do see this red color here. Again, there's over about I think about 60 different colors of quinoa that have been reported. So it's hard to say, dominant, recessive for each one of those, but we can come pretty close with our estimations. And then we'll take the population. So first of all, we'll grow out the F2 in the greenhouse, really just to increase the seed in a safe environment, so we don't want to lose any of that precious seed. So we'll increase it in the greenhouse, and then we'll grow the F3 to F5 populations out in the field. And so we're doing this in two mega environments of Washington State, the west side, so west side of the Cascades, and the east side of the Cascades. And the west side of the Cascades is much cooler, much wetter than the east side. The east side is hot and dry, and so they're very different. And so we're already, even though we don't do much selection at this stage, we do some random selection, but just the impact of natural selection is already affecting these populations. For example, last year on the west side of the Cascades, it rained quite a bit in August and September, about 4 to 5 inches. It's usually pretty dry over there, and so we had a lot of pre-harvest sprouting, and I'll show a picture of that later on. But the sprouting selection pressure was fantastic. We lost about 80% of our lines because due to pre-harvest sprouting. So the 20% we saved, we're really hoping that we can get some good sprout tolerance lines out of that with consistent sprout tolerance. And we think we can. One of the best parents for sprout tolerance is a variety called Redhead, which was bred and selected by Frank Morton of Wild Garden Seeds, and he selected that one after two very, very wet August and September fall harvest seasons. So we'll grow those out in two diverse environments, and then we'll start some advanced yield locations. So again, these are our three primary locations. This is on the Olympic Peninsula. We work with two farmers here. They get about 30 to 50 inches of rain a year, depending on which location they are, and definitely on the year. It can be more or less than that. And then, so again, this is where we test for sprouting tolerance, mold tolerance, that type of thing, but very little heat tolerance. So this whole area of Western Washington is ideal for growing quinoa based on kind of cool summer temperatures. We also are doing selection for heat tolerance here in the lower Yakima Valley. We actually do some heat tolerance selection here in Idaho as well, Lewiston, Idaho, where last year it got up to 107 degrees, and in our acre of breeding plots we didn't get one single seed. So that it was a little too hot to get any good selection pressure there, but we're going to keep trying. We are looking at irrigation and the effects of irrigation on heat tolerance as well. And then here in the Palouse, we've got about 1.5 million to 2 million acres of dry land wheat and barley production right now, some legumes, lentils, chickpeas as well. And this is where we do the bulk of our research. So Pullman is located here, and we've got trial farms all around this area where we're looking at dry land quinoa production and seeing how quinoa will fit into a common rotation on the Palouse, whether it would come after winter wheat or where it fits in, what it would replace in the rotation, or if it can just diversify rotation. One of the main groups of farmers we're working with too is a no-till or direct seed group of farmers. So it depends on the county. Whitman County, where we are, it's about 20 to 30% no-till growers. Down here, southeast, Walla Walla County, it's about 80% no-till growers. And so there are different traits that we're selecting within the no-till farming systems as well. In general, these are our traits of interest. They go beyond this. So nutritional value, that's kind of a broad one, but we have many different traits we're looking within nutritional value. Grain yields, very important. And then also early maturity, that's extremely important for us. Most of the varieties that we test, if they come from one of the main subgroups from Bolivia or Peru, they won't mature in time in our climate. So looking for early maturity is extremely important, as is heat tolerance. And then other things we're looking at: seed size, lodging resistance. Some of these lines grow 8 to 10 feet high, which is too high really for us because they start to lodge. So salinity tolerance we look at in particular with BYU and Utah State. drought tolerance. We have some insects that are a problem, lygus bug, aphids. Whether or not we see genetic resistance to these is still to be determined. Nitrogen use efficiency, saponin content, and sprouting resistance. Down here, this is a picture of Hannah Walters and Adam Peterson, two graduate students who have both just completed their master's and are still actually working with quinoa. So this is Downy Mildew. This is the #1 quinoa disease worldwide, a disease of quinoa. You can see the gray and pink discoloration of the leaves. This would be a fairly susceptible variety here. So this is something we're also looking at, primarily on the west side, but we also do see it in eastern Washington too. It does prefer cool, moist climates. You see, again, the discoloration, chlorosis, necrosis. It can drop yields down to 0% if it's severe. And this is something we're working very hard on here. We have seen it, and again, we're working with a lot of South American scientists who are also working on downy mildew resistance. Resistant varieties. This is one resistant variety. This one, again, this is cherry vanilla. This is from Frank Morton of Wild Garden Seeds, and because he's done selection of quinoa in western Oregon near the Willamette Valley for so long, he's already got some good varieties that are fairly resistant to downy mildew. So this is a picture of pre-harvest sprouting. You can see all the little sprouts coming out of the seeds here. This is quite common. We've just out of the last 4 years, this has happened twice to a majority of our seeds on the west side. So this is again genetically controlled and something we are I think on the cusp of figuring out, and any new varieties we'll release for western Washington might also become applicable to other parts of the U.S. as well, where there are those August summer rains. In addition to genetic resistance pre-harvest sprouting, we're looking at panicle architecture. So this type of panicle here allows a lot more wind to blow through it, and it keeps it dry, and we've noticed that these are often a lot less moldy than these ones here on the right. And it's really our natural tendency just to select these big, beautiful heads here full of seeds. But for western Washington or very rainy areas, these just tend to, the moisture just gets trapped in there, and over time will cause all of these to mold and or sprout. So these are the ones on the right side are better for drier areas, and then these ones on the left side are better for wetter areas. So saponins are really key in determining the processing capacity of quinoa. So I guess probably the major bottleneck we have in the U.S. right now is the lack of a processing facility to remove saponins. They've got really nice state-of-the-art facilities in Peru and Bolivia that do this through a combination of washing and abrasion, light abrasion. But we don't have that in the U.S. yet, and so we know there's a really big market for quinoa. Farmers now are realizing they can grow it with the right varieties, and they're figuring out how. So we, especially in this area, the Palouse, we know we can grow it. But we haven't been able to get that seed to market, and within the last month, we've heard that Andean Naturals, Sergio Nunez de Arco and Don Kukin are putting together a quinoa processing facility in the Sacramento Valley. So for us, for growers up here in Washington, it's about 4 to 7 cents a pound to ship it down to Sacramento Valley and get it clean. So that's going to make a big impact. One of the questions is, though, can we breed saponin-free varieties? And there are a few out there. They're patented. A couple we've seen growing in this region are from the Netherlands. They actually haven't done quite well in this area because I think they're not very heat tolerant. But it's a really interesting idea. So we have some saponin by saponin-free varieties or populations that we're developing right now, where we've made crosses and we're testing those out in the field. A problem with a saponin-free variety is that then you lose any benefits that the saponins impart upon the seed, and one of the main ones is that it is a bird deterrent and also has been shown to be an insect deterrent. But then on the other side, you do have a clean quinoa seed without the bitter soapy saponins on the outside. So our goal is to release both saponin-free quinoa varieties and ones with saponins on them as well. And then we'll let the growers figure out what works best for them. I'm not going to talk much about this now. We've got two students. This one here, Geang Wu, and here she's working on texture and micronutrient concentrations of different quinoa varieties. So really interesting research. And then Rocio over here with her PI, Juliana Norato. They're both from Peru but work at WSU. They're working on bioactive components and its effect on obesity in quinoa. So there's a lot of research we're doing there. This is all later stage selection. So once we're growing out 7 of our best lines just for both of these researchers to do some more work on, and if we can, then we'll release varieties that are more nutritious or have better heart healthy compounds or whatnot. There's a lot of different options there. So we are working with Brigham Young too on some quinoa mapping. They've done quite a bit of this, so our role is doing some phenotyping for these projects. We're hoping to identify QTLs for some traits of interest, as well as to develop varieties for our region. We've done a couple years of phenotyping so far. We've looked at 5 different populations, all coming from these 8 different varieties here. I just color-coded them to show you kind of the areas where they come from. The purple ones here are from more of the Altiplano regions, Salaris regions of Bolivia and Peru. So very high altitude, salt-tolerant, drought-tolerant, and they also have just very high levels of, or differences in downy mildew susceptibility. So those are 3 of our parents. When we grow them here, they don't do well at all. In fact, they don't even make any seed. And then we chose some parents from the Chilean coast as well. So these populations were developed at BYU and grown for about 7 years. So we've been testing the F7 and the F8 generations of these lines. And so these are from the Chilean coast. And when you see some of these, especially from down here, NL6, KU2, they make big, beautiful heads. And if, in good years, we get a nice harvest from them. These are what the parental lines look like when they're grown out in Pullman, so in the Palouse. NL6, these are a few of the other ones, KU2. And then these, so you can see the big head on NL6. When you look down here at Chucapaca, this is really what we get, a nice tall plant with some spindly, like finger-type branches coming out. And again, these won't make any seed. They'll start flowering in August, and by then it's too late to actually get a good seed crop for us for these. So these are the mapping populations we have. We don't have any data to share with you on these yet. Hannah just published her thesis, and we're sifting through all that right now to see where we are. But we grew these first 4 populations out 2 years in a row. This is the number of lines we grew out each year. This last year we had a really nice big population from this, we call it our GO population. And this is the only one with a Chilean cross with a Chilean. So we have great hopes that progeny coming from this population will actually be well adapted to our area. And if we're lucky, we might get something we can actually turn into a cultivated variety. So when we phenotype, we're looking at many different traits, but not all of them. Plant height and architecture are very important. Flowering, seed maturity, again, downy mildew. Leaf and inflorescence color, seed color, saponin content, protein content. And this is what we're looking at on almost all the progeny lines. When we get down to a certain point, then we can start looking at the nutritional value and other things that really we can't do on 800 different lines, but we can do on 10 or 20. So here's a picture of the one of the populations, and so here in this lower corner you can see one of the lines. It only grew about 18 inches, had nice big leaves. It might work out great for an edible leaf variety, but it doesn't make seed in our area, so that could be a drawback. But that's fine too. It'll make seed somewhere. And a lot of these are quite tasty. So right next to it, a sister line. This one grows to be about 9 feet, and again, this one didn't make any seed either. So there's a tremendous amount of genetic diversity within all these. You can see some downy mildew here on this leaf, and so within this is really our pool to select from, and what we're hoping for is something about 4 to 5 feet tall that matures by late August to early September, and is both heat tolerant, drought tolerant, and depending on where we grow them, also with a good potential for downy mildew resistance and pre-harvest sprouting tolerance. So I'll end there, and hopefully we will have time for questions.